Welcome. Are you taking AP Biology? Well, if you are, you're in the right place because today we're going to be covering Big Idea 1, all about evolution. Sorry for technical difficulties. We were having a little bit of issues, me getting logged in. I'm so sorry, y'all. I am Tiffany Jones, and I come to you from Georgia. I run AP Bio Penguins, and so I decided to collaborate with Market Learning. Well, they're collaborating with me so that I could bring this review session to you. So real quick, you're probably wondering, well, why am I calling these kids AP Bio Penguins, right? Well, because penguins are just success. So since you're sitting in this review session, you are now an AP Bio Penguin and you are dressed for success. So now there's a little bit of a delay. And in the past, I've always had a moderator like kind of running the chat for me. So if you type anything in the chat, like I will see it, but it's like a 30-second delay. So by the time that I see it, respond to it, it's like a minute delay. So there might be some like awkward times where I just answer a question randomly. It's because I'm seeing it in the chat, but I will answer your chat questions as we go. So today's plan is that we're going to do Big Idea 1. Now, I've never taught them by the big ideas, so I'm doing my best as we work through this. Thankfully, this big idea is mostly Unit 7. So we're going to do a little hair in Unit 2, just about the origin of cell compartmentalization. We'll do a little bit in Unit 5, just talking about heredity in terms of just DNA and RNA. And then we'll do a little bit in ecology, just talking about how disruptions in the ecosystems can lead to different changes within the environment, which then of course causes changes in terms of evolution. And then we'll hit a whole bunch of Unit 7, which is all about natural selection, which is the bulk of evolution. So, introduction, natural selection, artificial selection, population genetics, a little bit on Hardy-Weinberg, evidence of evolution, and then common ancestry. So because of doing it this way with the big ideas, there tends to be a lot more content, a lot more meat. I have some questions built into the end. I'm going to hopefully get to those, but if I don't, sorry, love you, please don't hate me. So real quick, let's go to Unit 2. So this is the origin of cell compartmentalization. So first thing, let's think about, there are prokaryotes and eukaryotic cells. So what is the big differences between a prokaryote and a eukaryote? So a prokaryote has no membrane-bound organelles. So there's no Golgi, there's no ER. They don't have any of the organelles that you all had to memorize in Unit 2, okay? But what they do have, they have ribosomes, they have a plasma membrane that will surround it. They have DNA, that DNA is in this nucleoid region. And the thing about their DNA is that they only have a single DNA, like there's one DNA, and it's circular. And then it also lacks histones and it lacks introns. And so if you remember, histones are going to be the proteins that DNA wraps around. It's a way to organize DNA in eukaryotes because of the fact that they have so much DNA, they need some way to organize it. And then they also don't have introns. Introns were the things that were removed out. It's like the non-coding content that was in the DNA that was cut out. I'm sorry, not the DNA. The non-coding genes that were in the mRNA that's cut out of the pre-mRNA before it leaves the nucleus. Well, as we're looking at a prokaryote, there's no nucleus. So all of the genes have to be ready to be translated because oftentimes they're transcribed and translated simultaneously. So they do not have histones or introns. Now, when we think about eukaryotes are significantly larger. They're a lot bigger. They have membrane-bound organelles, so they have that nucleus, they have the Golgi, they have the ER, they have the peroxisomes, they have all of these different organelles all in them. Their DNA is multiple. They've got more than one strand of DNA. For example, humans have 46 different strands of DNA. It's linear, so it's long strings of DNA versus, as we saw, prokaryotes were circular, so kind of circles. And then also it does have the histones to wrap and organize, and it also does have the introns that are removed. That allows for that alternative gene splicing when we talk about that in, Unit 6. So how did we get these membrane-bound organelles? How did we get all this stuff in there? Well, there's something called the endosymbiotic theory. And so the logic is, or the theory is, that back in the day, there was a little bit of invagination. So there was some infolding that happened, and that infolding created a lot of the membrane structures that we saw inside the cell, okay? So after it did that, they had this like ancestral cell, and that ancestral cell engulfed a prokaryote. So when it engulfed it, they create a symbiotic relationship. So they both benefit. And so that prokaryote that was engulfed was a heterotroph. So it was able to use chemicals, organic chemicals, break down those organic chemicals and be able to produce energy or make ATP from that. And so we found that, of course, that must have happened first. The reason why we think that the heterotroph was engulfed first was because of the fact that all eukaryotes have a mitochondria. And then there must have been some type of divergence. There was one of these cells that engulfed a photosynthetic prokaryote. And would engulf that photosynthetic prokaryote, that led us to this, pathway that has all the plants and the other organisms that are able to photosynthesize. And so we say all this, but do we have any evidence for this? Well, there's a couple different pieces of evidence that support the endosymbiotic theory. So the first piece, the DNA, number one, there is DNA in mitochondria and chloroplasts, and that DNA is circular. And as we mentioned, a prokaryote has circular DNA, so that kind of tracks. Also, the DNA lacks histones. The mitochondria is able to self-replicate. And that self-replication is done by binary fission. So it goes through the same division process that a prokaryote goes through. The ribosomes that it has are similar to prokaryotic ribosomes. It has a similar inner membrane that's similar to the prokaryote. So if you would think that something has engulfed it, right, the own membrane of the ancestral cell is now on the outside. And so there's double membranes and triple membranes that make up the mitochondria and the chloroplast. So that also kind of provides a little bit of evidence for us. The mitochondria and the chloroplasts are able to go through transcription and translation. They're not requiring any type of proteins from the eukaryotic cell. And they're approximately the size of a prokaryote. And then they also have prokaryotic-like enzymes. So there's a lot of evidence that kind of supports this. So that's all we need to talk about for Unit 2. So we just had 2.11. So going into Unit 5, we have to talk a little bit about mammalian genetics. Specifically, we're talking about just DNA and RNA. So all organisms have DNA and they have RNA. This supports that the prehistoric, ancestral cell had DNA and had RNA. And so as a reminder, DNA and RNA is made up of pentose sugar, it has nitrogenous bases, and it of course has the phosphate. That's the three things that make it up. DNA has deoxyribose, RNA has ribose. Nitrogenous bases we have are adenine, thymine, cytosine, guanine, and uracil. Remembering that DNA has thymine, uracil is in RNA, the other three are in both of them. I think you kind of remember most of that. Also, think about the genetic code. So all organisms, they have ribosomes, and they're able to use those ribosomes to synthesize proteins. So you can take a human genome, let's say, I don't know, the gene for insulin, and you could incorporate that into the genome for a bacteria. And now that bacteria can make human insulin. So the code is universal. You're able to take, the same code from one organism and put it into another organism, and it can read that code and appropriately synthesize the product from that code. That shows us that there is common ancestry. That shows us that we have this common ancestor from, common descent, and that we're all from the same spot. And then metabolic pathways. There are core metabolic pathways that are common between multiple organisms. Glycolysis, the creating of carbohydrates is done in the same way in all cells. Synthesizing phospholipids, so all these core metabolic processes take place in all these organisms. And you're welcome to type stuff in the chat as we go. I can see the chat, y'all. Hopefully everything's going good, but y'all can hear me and we're good. Otherwise, I hope you would have told me by now. So let's think about the central dogma for a second, because we just mentioned that we have DNA and we have RNA. So DNA to RNA, that process is called transcription. RNA to protein, that process is translation. Well, there are things like viruses that can ignore this, that can do something different. And so specifically retroviruses. Retroviruses like, I don't know, HIV, they're able to use their RNA genome and they can go through reverse transcription. They use an enzyme that they have called reverse transcriptase. And that enzyme allows them to use the RNA as a template to synthesize a DNA strand. So it makes, an RNA DNA hybrid and then makes a DNA. And then it's able to incorporate itself into a human genome. And so then it goes through the normal central dogma. So this central dogma kind of brings us back to the last universal common ancestor, Aluka, which I was talking to someone last night trying to work through the PowerPoint and try to get my mind wrapped around the big ideas. And she's like, well, just talk about Luca. And then my mind just immediately went to the Disney movie. So if your mind just went to Disney movie, Luca, you and me both. Anyway, so this was our last universal common ancestor. It's the most recent common ancestor of all life on Earth. So it doesn't mean it's the very first cell, but it's the first cell that we've all kind of diverged from. And so it shows because of their shared modern genomes. They also have the genes that are needed for transcription and translation that was needed to convert the DNA to the RNA to the proteins, which is all part of that central dogma. So my system keeps glitching. I don't really know why. I can't get this slide to fix. I worked on it for 10 minutes before, but I just gave up. So unit 8, we have one little section to do here. This is disruptions in ecosystems. So kind of how things can change in the ecosystem can, of course, cause changes in evolution and our organisms. So first thing, adaptations. So because of something that might happen in the environment, the organisms are going to have to be able to adapt. And when I say the organisms, I'm talking about a population of organisms, because individuals do not change, they do not evolve. So the individuals that are better suited for the environment are going to be more likely to survive. And so as we have these changes that might take place in the environment, certain organisms are going to be favored. Certain individuals will have that favorable trait that will allow them to be more likely to survive. And if they're more likely to survive, they're more likely to reproduce and more likely to leave more offspring. And that provides them, of course, as we said, it gives them an advantage. So here we kind of think about a giraffe. So back in the day, giraffe had a short neck, but there was a mutation that happened. And because of that mutation, the giraffe got a little bit of a longer neck. And so because of the longer neck, because it could reach more food in the trees due to its mutation of the longer neck, then it was able to get more food than the other organisms. So it was more likely to survive. And of course, more likely to reproduce and more likely to leave more offspring. So that leads us, as we said, mutations. What is a mutation? A mutation is just a change in the DNA. Okay. Now it's random. As DNA polymerase is making copies of the DNA, it makes an error. Just like as you're typing on your computer, or you might be taking notes right now, you're going to accidentally write something down wrong. You'll use your eraser and you'll erase it. But sometimes you don't catch your error until afterward. And that's what's happening with DNA polymerase. As it's base pairing, it makes an error every once in a while. And so that error sometimes just goes on. And so it's not like it's picked for a certain environment. The mutation that happens just happens to make the organism more likely to survive. And because it's more likely to survive, of course, we already said the rest of it. Okay. So Industrial Revolution. Back in the day, we had mostly these peppered moths that were kind of a lighter shade. And as you notice, the tree was also a lighter shade. But because of the Industrial Revolution, because of all the guck that was put up into the atmosphere, it caused the trees to get darker. So there were some moths that had a mutation. That mutation caused them to be darker. And because of that mutation, you saw they could blend into the trees. And so we saw a change in over time of the population of the lighter peppered moths and the darker peppered moths. So you can kind of see that they're, actually opposite of each other. Yay. So all that was the unit 2, 5, and 8 that kind of corresponds. Now we're really just jumping into unit 7, and we're just going to stick in unit 7 for the rest of this. So 7.1 is Introduction to Natural Selection. It was developed by Charles Darwin. He got on a boat. He traveled on the HMS Beagle. He ended up in South America. He was looking at all this stuff. He's like, oh, cool. Hey, I see different organisms. Okay. So due to all of his observations, he came up with this theory. I know you've all heard it. Oh my gosh, my brain. The survival of the fittest. But of course, we as AP Bio students do not use those terminologies, right? Because that is not good to use on the exam because of the fact that what is fitness? So if you think about fitness and you ask a person, hey, what's fitness? They're probably going to tell you the person who is the strongest, the person that's the healthiest. They're going to tell you all this stuff. But in actuality, fitness is just your contribution to the genetic pool. So the more fit you are just means you're having more offspring, and those offspring are able to also survive and be able to leave more offspring. So that's what he means by survival of the fittest. The organisms that have the favorable traits are more likely to survive and pass on those traits. And so in order to do that, we have to have competition. There has to be some type of fighting for resources. As we saw with the giraffe with the longer neck was more able to get the food. And because it got more food, it was more likely to survive over the other organisms or the other giraffes with the shorter necks. So kind of thinking about that competition for resources, okay? And as we already said, more favorable phenotypes are going to be more likely to survive. And so evolutionary fitness is measured by that reproductive success. It's measured by the ability to reproduce and be able to pass on those favorable traits to the next generation. And also, just because something is favorable now doesn't mean that it's always going to be favorable. As the environment is constantly changing, these favorable traits are going to change in that environment. As we saw with the peppered moss back in the day, a light shade was favorable. But as the environment changed, as the trees got darker, the darker phenotype was more favorable. So as things change, of course, those organisms, those populations are going to have to change with it. So here we can kind of see that natural selection. Those individuals that were darker are more likely to kind of blend in. And so we can kind of see a change. Y'all don't know what the A and the little a mean in terms of like the research that we're, not research, but the review that we're doing. So just as a reminder, capital A is your dominant trait. So it's the frequency of the dominant, and little a is the frequency of the recessive trait. And we're seeing that, if those traits, those frequencies change over time, that means evolution is occurring. That goes to Hardy-Weinberg, which we'll talk about in just a little bit. So examples, we already talked about the peppered moss, but let's talk about antibiotic resistance. So you're sick, you take an antibiotic. So the antibiotic's job is to kill the bacteria. Well, it just happens that some of the bacteria have a mutation. And because of that mutation, they're more likely to survive. They can evade this antibiotic. And so we're going to see that over time, because of the fact that they survived, we're going to see an increase in the resistance of the bacteria that are resistant to antibiotics. And that's why we've kind of seen this new issue of antibiotic resistance. And that's why if you have little siblings, you notice that they change the antibiotic every time your sibling gets sick. And so that they're able to try to get the bacteria cleared out, because hopefully it's not resistant to everything. So when we think about this, we think about there's all these factors. We've already talked about biotic and abiotic factors. Biotic factors are just living things that can cause individuals more likely to survive or less likely to survive. Abiotic would be non-living things. We saw the darkness on the trees. And so different genetic variations are going to be selected in each generation. So over time, as the environment changes, different traits are more favorable or less favorable, which is what's going to lead to that difference in our variation. And then I also just want to give a quick caveat to be very careful of Lamarckian statements. So some of you may be saying, oh, well, the giraffe has a long neck because it stretched its neck. Well, true, you stretch your neck, logically it might get longer. Yeah, but that's use and disuse. That's what happened during your lifetime, okay? You don't pass on a trait to your offspring unless it is in your DNA. So you stretching your neck doesn't change your DNA. It's just you using a certain part of your body a little bit more. And so be very careful about Lamarckian statements and saying that because of, this organism doing this thing, it's going to, pass on those traits. And same thing also, don't say that the antibiotic made the bacteria resistant. It provided a selection in the environment, which then of course caused natural selection to take over, and the antibiotic resistant bacteria were more likely to survive. So you got to be very careful about Lamarckian statements. So 7.2 is also a natural selection. So let's just real quick talk about the fact that natural selection is going to act on a phenotype. Nature doesn't know whether you're homozygous, dominant, or heterozygous. It just knows what trait you have. You have sickle cell, so there's low health care here. You're less likely to survive in this low healthcare area. You are homozygous dominant, you get malaria, you're more likely to, not survive, but you who heterozygous can survive, right? So you've got this trait. And so natural selection can only act on the trait. And then as it acts on that trait, it changes the genotypes. It makes it so that a certain genotype is more favorable or less favorable. And I feel like I just talked in circles, so I'm so very sorry, y'all. So these different phenotypic, these different variations are just coming from mutations. We need there to be variation in the population. The more variation in the population, the higher the chances that if a random thing happens, it's not going to wipe out your whole population. Thinking about the bacteria is all the exact same, other than a couple of them that have mutation. For the most part, they're all the same. They don't go through the meiosis process of crossing over, independent assortment, all the other things that provide for genetic variation. They don't do that. And so if something happens, it was going to wipe out the whole population theoretically. Okay. So some traits are going to increase the fitness. So as you see, then this mouse that's lit on the sandy background, it's more likely to survive because the hawks can't see it and eat it. And same thing here, this mouse can blend in. So of course, these traits are increasing their fitness. But these traits, if you put the mice in the opposite background, they're less likely to survive. And so you're seeing that this is going to actually decrease the fitness. And remember, fitness just means that it's a contribution to the next generation is contribution to the gene pool. So when we talk about natural selection, there's different things that can happen. You have disruptive selection. In disruptive selection, you're going to select for the two extremes. I don't know, Darwin's finches, let's say that there's a drought. And so there's only like these itty bitty little fruits, and then these are really big seeds. So the bird that has a small beak can eat the little nuts and berries. They're perfectly fine. The bird with a big beak can get to these hard shells, crack them open, all that fun stuff. But the bird that has a medium beak can't get food. Like it can't compete with the small bird beak. It can't compete with the large beaked bird. So it doesn't have food. So we see a decrease in the intermediate and an increase in the two extremes. Stabilizing selection is where we're selecting for the intermediate. The traditional example we see here is on birth rate. If the baby's born too, like, again, we're talking about like before modern medicine. If the baby was born too small, then it's probably not going to survive. If the baby's born too big, probably also not going to survive. So we found that birth rates are kind of, I don't know, between like, I think it's 6 to 8, 5 to 8 pounds is like the traditional baby birth weight. And you can kind of ask your parents later on what your birth weight was. And then directional selection is where it's going to move in a direction. It's moving toward one of the phenotypes. So we saw with the peppered moths that as the Industrial Revolution took place, the colors changed from a lighter shade to a darker shade. So it moves in a direction. So that leads us into artificial selection. So artificial selection is logical, right? There is something, a human, that's selecting for a certain trait because they find that trait to be favorable. So it's not because it makes the animal, more favorable in some way. It's because we as humans like a certain thing, and so we want more of that thing. And so the big examples we kind of talk about would be, of course, dog breeds, right? So dogs all came from a wolf. That was kind of back in the day. And then they were domesticated, and there were certain traits that we liked in them. Bloodhounds are good for hunting. My mind is currently going blank, but there's all these different specific traits that we have for dogs. And that's because of artificial selection. We've selected for those traits and we've bred those organisms together so we can kind of have more of that trait in that organism. The reason why we have mules is because of the fact that they've got the robustness of a donkey, but they're, the rigor of a horse. Like, so, we are doing this, crazy people. Corn came from maize. So back in the day, corn was like this itty bitty little maize. And now we've got nice little plump corn kernels that we love to eat. And the wild mustard, different components of it were used to make different vegetables that we eat, like broccoli, cauliflower, kale, cabbage, and I cannot try to even pronounce that last word. So convergent evolution is also what we kind of talk about in this section. I don't know why it's connected here, but this is where it was in the standard, so this is where I'm doing it. So convergent evolution has to do with that you have a similar environment, which leads to a similar selective pressure. And so because we have this same pressure, it's going to select for the same trait, even though the two are not related. So my favorite example is the sugar glider. Okay. So the sugar glider is a marsupial. It's in Australia. And well, actually it's supposed to be in Australia, but people I think have bought it and it's all over the place now. Anyways, so the sugar glider is a marsupial. It has a pouch, just as you've seen with a kangaroo and all that fun stuff. And so it has a early premature birth. So remember that. The flying squirrel is, from North America, and it is a placental. So it is going to grow to full term in utero, and then it's born at full term. So you can see right there off the bat, they are completely different. But notice they both have this little extension underneath their arms. That little extension allows them to jump off a tree and kind of glide to the ground. So they have a similar adaptation to a similar environment, and they are not related. And so oftentimes they'll ask you questions about whether two organisms are related, and you kind of have to think about, are they related by a common ancestor? If there's no common ancestor, we're looking at convergent evolution. Same thing as a bat wing and a bird wing. Okay, a bat wing and bird wing are not. They are again similar structure due to a similar selection. So population genetics. So what are we talking about here? This has to do with the different mutations and different DNA traits that we're going to see. Okay. So reduction of genetic variation within a given population is going to increase the differences between the populations of the same species. Okay. And so what we're looking at in this section is how the genotypes can change and how the different gene pools can change. So first thing, mutation, we already mentioned before, is a change in DNA. It's random. There's no selective pressure that causes it. It just happens on its own as DNA is being replicated. But this is what leads us to the different variations that natural selection is able to act on. It's what's giving us the different phenotypes that you may physically see that may make one individual more likely to survive in that population. So genetic drift is random, and it is going to change our allele frequency. So the first one we have is a bottleneck effect, which I was having issues with the linking, so I'm sorry that my picture just did crazy stuff. So with the bottleneck effect, there's some type of natural disaster. That natural disaster is going to wipe out some of the individuals. Now it's random which those individuals don't survive, okay? So it's kind of like taking a bottle and like turning it over and like shaking out some beads that are in it, okay? So the new population is not representative of the original population. It doesn't have the exact same allele frequency. It is a random thing that happened that changed our allele frequencies, which isn't really, whatever. Founder's effect is where we see a very small population that gets separated. So as you see, like these little lizards, they somehow, drift over to this island and they create a whole new population on that island, okay? That new population is due to the new population is not going to have an allele frequency that is representative of the original because it's random which of those lizards went over to that island. So these are different random things that can happen that will reduce our population size and it will decrease the genetic variation. And this can lead to some of our harmful alleles becoming fixed. And so those of you who don't remember what fixed means, it just means that all the individuals are of that certain genotype. So I tell my students, if we look around our classroom and we all have 5 fingers, and you can kind of look around your own classroom and see this too. Having 6 fingers, being polydactyly, is a dominant trait. And so since everyone in my class has 5 fingers on both of their hands, we know that we're homozygous recessive. So that allele is fixed. Every single one of us have a recessive allele at every single one of our chromosomes, like our chromosome, whatever chromosome that allele is on. So there's that. Okay. And then the last thing we talk about is gene flow. Gene flow is just immigration or emigration. So how individuals are moving in or out of a population. So when they move into that population, they're going to potentially bring in new alleles. This happened with the DDT. So DDT is was kind of used to get rid of mosquitoes and other insects. And so since those are insects, of course, they could fly. And so there was a population of insects that came in the area that were resistant to the DDT. And so they, of course, more likely to survive. And that whole now population became resistant also because the immigration of a mosquito that had that allele. We also see immigration. They could, of course, move out of the population. This brings new in, removes alleles from, all that fun stuff, which is going to change our allele frequencies, changing, of course, our genetic variation. Whew. On to Hardy-Weinberg. And y'all, you're welcome to type anything in the chat if you have questions on anything. I see, there's only 7 of you in here. I'm so happy you stuck with me. And I hope that this has been helpful. If you need anything, just type it in the chat. So Hardy-Weinberg equilibrium is a mathematical formula that we're going to use in order to say that yes, evolution took place, or no, it didn't take place. And it all has to do with our allele frequencies. Okay, so there's 5 conditions. And if you want to watch a good video that'll kind of help you out with this, it's a TED Ed video. It's called The 5 Fingers of Evolution. And so it walks through the 5 different conditions. And I may do them differently than what's on the board because I'm going to do the actual 5 fingers. Because of the fact that this is in the order of, why is it popping up like that? Go away, machine. This is in the order of the video, okay? So small finger has to do with that we want there to be an extremely large population size. If you have an extremely large population size, then these random genetic drift situations are not going to affect your allele frequencies, okay? We want there to be random mating. And usually I have my my wedding band on, but I forgot it as I was rushing to get upstairs. I'm so sorry, y'all. But so like we usually think about like a wedding band worn on this hand. And so that's going to cause us to think about, okay, well, we need there to be random mating. You can't mate with a certain individual because of certain traits, like the blue-footed boobies that have the beautiful blue feet. You're not going to like pick them because of their beautiful dancing number that they're going to do for you, right? The middle finger for no mutations. We want there to be no mutations. A mutation is going to add a new allele into our population. It changes, of course, our allele frequency because of that new allele. Pointer finger is to kind of think about movement. We want there to be no gene flow, no immigration, no emigration, and of course, no natural selection. No trait can be more favorable or less favorable than another trait. All traits are just equally favorable. Yay, that's the 5 conditions. So if all those are met, then theoretically, evolution is not taking place. And so again, check out that TED Ed video. It's got Paul Anderson, the guy that does boson biology, which you should all be watching because he's amazing too. And then so from that, we're going to say, okay, well, did our population evolve? And that's where we're going to look at the allele or the genotypic frequencies. If either of those change, that means that the population has evolved and evolution has occurred. So the different equations we're going to use, there's 2 different ones to use, and there's different times to use them. So these are your different variables you have in them. P is going to be your dominant allele, or the frequency of your dominant allele, and then Q is going to be the frequency of your recessive allele. Okay, and so some students are always like, well, Ms. Jones, how can I remember that? And I'm scrambling because I'm trying to get a piece of paper together. So the way I tell my students is, is my camera flipped? I think the camera flipped. Anyways, so P, okay, if you flip it upside down, it makes a D. And I think my camera's backwards, so it looks like a weird letter, but I tried, okay? Q, as we said before, frequency of our recessive allele. P squared, so having 2 of our dominant alleles, we know is homozygous dominant, so the frequency of the dominant allele. Q squared, 2 of the recessive alleles, so of course homozygous recessive. And then 2PQ is 1 dominant and 1 recessive allele. And so if you were to draw it on a Punnett square and do PQ and PQ, you would see this whole equation that we're about to see in a second, which is that P plus Q equals 1. So the frequency of the dominant allele, frequency of the recessive allele is going to be 100% of the alleles. And then the frequency of homozygous dominant, frequency of heterozygous, and frequency of homozygous recessive should also be all of them, so 100%. And so I use this little nifty chart to help my students out. And so I tell them that they need to draw this chart every single time that we do a problem. So you're going to start with Q squared. Always start with Q squared. Did I say that loud enough? Again, always start with Q squared. Okay, so if we know Q squared, the way that we can get Q, if you take the square root of it. So we take the square root of Q squared, which gives us Q. We know that P plus Q equals 1. So 1 minus Q gives me P. And then I know P, I can do P squared. And I know P and Q, so I can do 2PQ. And so that's one way that you can solve a Hardy-Weinberg question, okay? You're gonna solve it this way. If they just tell you, 50% are this genotype, or 50% are this, okay? Now, if they tell you there's, 200 that are homozygous dominant, 400 that are heterozygous, and I don't know, 20 that are homozygous recessive, they give you specific numbers for each of your different genotypes. You need to count the alleles. And this equation is funky. I've written this equation. I just made this thing up. I don't know what the formula equation is. This one is not on your formula sheet. The last one is on the formula sheet, but this one is not. Okay. And so it's just a matter of counting the alleles. So I just made up my own little formula to try to help you out. Okay. So homozygous dominant alleles are going to have 2 of that capital A, that dominant allele. Okay. So we'll do 2 times the number that are homozygous dominant plus the number that are heterozygous. Because a heterozygous individual is going to have 1 of the dominant alleles. And then you'll just divide it by your 2 times the number of individuals. And I'm going to answer that question in just a second. So 2 times the number of the individuals. And then Q is going to be 2 times, of course, your homozygous recessive plus your heterozygous. Because again, we're counting the number of recessive alleles in the population. And then you'll divide by 2 times the number. And then P squared is just number of homozygous dominant divided by the number of individuals. Heterozygous is, number of heterozygous by the total in the population. And then Q squared is, of course, homozygous recessive individuals divided by total individuals. And someone just asked, are we going to be doing more of these sessions? Of course, I am doing 60 sessions, including this one. The next one will be Big Idea 2, and we'll go all the way through 4. And then the 2 that we'll do in April are going to be on the FRQs. And so it tends to be the second and third week of the month. So I'm doing one next Saturday, and then I'll do another one on March 11th and March 18th. And then it's weird week. I don't know what weeks they are in April, but check it out later. I'll figure out. Okay, so let's do some example problems. So Gardner School always has red, pink, and white snapdragons. There's 200 red, 300 pink, and 500 white. Determine the allele frequency of the flower allele color. Okay, so they're telling us the number. So we know, gotta use the allele method. Okay, so we pull up our alleles. Okay, so it said that we had 200 that were red, and I know there's 1,000 individuals. So that means that p squared is 0.2. 300 that are pink divided by 1,000, giving me, of course, 0.3 for my 2pq. And there's 500 individuals that are white, divided by 1,000, giving me 0.5 for my q squared. Okay, and then to find my p and my q, I say 2 times 200 because there's 200 individuals that are red, and they have 2 of the red allele. And then divided by 2,000, plus 300 because there's 1 red allele in each of those. And then the same thing would apply with our q, is that we would take 2 times the 500 because there's 500 that are white, plus the 300 that are pink because pink only has 1 of the alleles, white has 2 of the alleles, giving us our p and our q. So if a population that's in Hardy-Weinberg equilibrium, 36% of the individuals have the dominant phenotype for a certain trait. And so they tell you dominant trait. Well, dominant is p squared and 2pq, right? You all know that in complete dominance, having homozygous dominant and having heterozygous means that they both have the dominant trait. So you have to do a little bit of thinking here. Again, I told you to always start with q squared. We always start with q squared. So what I've given you is not q squared. So you'd say, okay, well, 100% minus 36% will give me 64%. That is your q squared. And that's what we're going to solve using. So we have our q squared of 0.64. Take the square root of 0.64, you get 0.8. Say, okay, well, 1 minus 0.8 gives me 0.2. And then 0.2 squared is 0.04. And then, of course, 2 times p times q. So 2 times 8 is 16. So I've got 2 times 0.16 gives me 0.32. And so that is how you would fill this in. And then I tell my students to fill the whole chart in, and then afterward, figure out what question they're asking you, figure out what they need. Because sometimes you just kind of have to work through it all first. That's just my thought process. So 7.6 is on the evidence of evolution. So there's a lot of different pieces of evidence we have for evolution. First thing is geographical. So we can see, okay, well, these share similar genetics, and they're located in a similar environment. So these have all, evolved from a similar salamander species. You can kind of see that they've moved up the coast of California over time. This is also leading us into kind of a speciation. These are different like subspecies of each other. It's kind of interesting lab. I have my students do it every year where they have to like graph where they all are. They hate me, it's right. So geological fossil evidence. So we can use different fossil evidence to see where individuals are. We can also use biogeography and see, based on, the geological time period, where were they? And you can kind of map out where these organisms were from their fossil evidence, and you can put Gondwana back together. It's pretty cool. So how do we date fossils? Well, we're going to date them either using, where was the fossil found? So you know that the lowest level we have are going to be your oldest fossils, and the highest fossils are the most recent and the newest fossils we have. You can also use kind of radioactive decay. So use the half-life. So half-life is just the amount of time that it takes for the amount you have to split in half and be half as much. Geographical data. So of course, biogeography, looking at where the different individuals are to be able to date them from, oh, well, you were near this thing back in the day. And then they also use kind of homology. So you can use morphological evidence, and you can use vestigial structures to see kind of how these organisms are related based on their fossils. And there was a question back in, I think, like 2014, 2015, where you had to figure out where the transitional amphibian species was, and it gave you different layers of fossils, and you had to pick which one it was. It's kind of cool. You can also use mathematical data. So we already talked about Hardy-Weinberg. So I just put a little graph up here to help us to remember Hardy-Weinberg. And this graph has kind of shown up on the exam as like a numeric response. There's no numeric response anymore, but that doesn't mean like this kind of graph can't show up again. So you can kind of see that, p squared plus 2pq plus q squared is going to equal 1. So you can kind of see kind of how the allele frequency is changing over the time. And then also you can see the P and the Q and what those ones are. And so they ask the individual, like the students, to read it and say, okay, well, at this Q squared, what is going to be the P? And they had to use the graph to find it. Yes, you could mathematically do it, but they asked the kids to find it using the chart. Biochemical is going to be using DNA or proteins. And so you can compare the DNA and see how many differences there are in the DNA. You can compare the proteins and see how many differences there are, of course, in the proteins. And so here you can kind of look at the human, the gorilla, and the chimpanzee. And you can see that the human is closely related to the chimpanzee because they share all of the same amino acids in this sequence for their hemoglobin, versus the gorilla is more closely related kind of to, maybe the chimpanzee. It has one difference here. And then you can see the horse and the zebra are closely related because they share all the same amino acids except for that last one. So oftentimes they may ask you, which one's better, DNA or proteins? And so you kind of have to think to yourself, well, DNA can have changes, silent mutations. And from those silent mutations, you could have a change in DNA, but not a change in the amino acid sequence. And so DNA is always the better option in terms of which one is more accurate, because you can have silent mutations that take place. It could also take place in an intron that gets removed. It could be taking place in a non-coding region. And I'll answer that question in just a second. Also kind of looking here at our amino acid chart, this looks at different organisms. And there's traditionally a phylogenetic tree or a cladogram on every AP exam, all the ones I've seen at least, where you have to draw it. And so using this is important to be able to understand. So you kind of see where they interact. So for example, E. ferris is going to have 0 differences with E. ferris because they're the same organism. That's why there's this big line of circles going down the diagonals of zeros. So what they're asking you to do is find out, okay, which two are most closely related. So a small number of differences shows that they're closely related. So the E. ferris and the E. africanus are closely related. And then the A. forestria and the G. gallus is also closely related to each other because they have 3 differences. And that leads to the D. polyleptus to be an outgroup in this example. And so you kind of have to use kind of where they intersect to try to help you to understand that. And first, just ask in the chat, do these live streams get saved on this channel? Yes, they get saved on the channel. And then I also will post them to my website. So the last piece of evidence we have is our physical evidence, looking at different homologous structures. So you can kind of look at that they have the same bone structures because they have a common ancestor. So here you look at a human, a cat, a whale, and a bat. They all have the exact same humerus. Now that humerus has changed. It has evolved. It has, for the different functions that they have in different organisms, but it is the exact same bone. Same thing as our radius and our ulna radius are also common. The metacarpals, our carpals, and our, of course, phalanges. And notice that's like a bat wing is like all that. So cool. Anyways, vestigial structures are going to be anatomical structures with no function. Think about your tailbone. Your tailbone has slight small function, but it's remnants from our ancestors. Our ancestors actually had a tail. And so if you look at embryonic development, you can actually see that we all have a tail during embryonic development. So at early phases of development in utero, there's a tail that forms. And then apoptosis takes over and we lose our tail. But there have been cases where individuals were born with their tail because, whatever. And so that's just a vestigial structure. It's not really needed. Another thing would be goosebumps. Our retroculi muscle kind of back from when we used to have fur, and the fur pumping up would keep us warm whenever we had, when we were cold. So that's kind of also another vestigial structure. So we also need to be careful about analogous structures. Analogous structures are due to convergent evolution. We mentioned convergent evolution. That was the sugar glider and the flying squirrel, right? They both had the little extension underneath their arm that allowed them to glide from the trees. So that's an analogous structure. They're not related to each other, but they look similar. So you want to go, oh, those are related, but they're not, okay? So 7.7 on common ancestry. So evidence of our common ancestry. All eukaryotes have membrane down organelles. Yay. We all have linear chromosomes. And then we all have introns. So these are the big three that were on the curriculum and on the CED. And I tried to stick as closely to the CEDs as possible when I was doing this PowerPoint. I really didn't have much to add to that, except for these are the three things that we have in common in terms of all eukaryotes. And that's why we show that there's common ancestry. So 7.8 is on continuing evolution. So evolution is a continuous process. It is constantly changing. As the environment changes, individuals are going to be more favorable or less favorable. So we find that the populations are constantly changing. You will never be perfect, okay? So genomic changes happen over time. If you look at the fossil record, there's continuous changes in our fossil record as these different bones have been changed over time, as they provide new functions, as they provide organisms to be more favorable. And this is also where we see the evolution of pesticide resistance, antibiotic resistance, herbicide resistance, and chemotherapy drug resistance. That's why our HIV drugs have to be cocktails. They have to be multiple different drugs that affect different parts of HIV in order for them to be successful at limiting the transmission rate of HIV. And then pathogens evolving and causing emergent diseases, okay? So here you can kind of see the increase of antibiotic resistance. It's just showing you the resistance of the bacteria. I was just using it as a graph to kind of show you that resistance has increased over time. And then thinking about emergent diseases, this is a graph I found about COVID and all the mutations that are taking place in COVID. And it's because of the fact that it's an RNA virus, it's constantly making errors. You can kind of see all the different lineages that are taking place from this one viral material. And so this shows evolution continuously happening. So 7.9, phylogeny. There's always a phylogenetic tree, always a phylogenetic tree on the AP exam. So what is it showing? It's showing us our relationships. It's showing us, it could show the amount of time, it's like amount of changes happened over time. It could show us how much time there is. It could show us, number of differences. It all depends on which one they give you. And you kind of have to look very closely. It can show characters gained or lost. And it can also show us our recent ancestor. So a cladogram is going to be like this V-shaped thing. The words are actually used interchangeably. So here, this was a question from, I think, the 2018 exam. They, of course, made you do a phylogenetic tree. I'm sorry, cladogram. And so using this, we can see we have a panda, a black bear, a brown bear, and a polar bear. And you can see that the black and brown bear have one nucleotide difference. So we were going to put those closely together. And you can see that the panda has the most differences between all the organisms. So we will put that at this far one. So it should go brown, black, polar, and panda in terms of our order. Okay. Now notice that the black and the brown bear have this node. You could flip the black bear and the brown bear, and it was going to be the exact same. And you're perfectly fine to flip those either way. If there's a node, they can rotate. So outgroup is going to be the organism that is least closely related to the rest of the organisms in the phylogenetic tree or the cladogram. And so, for example, panda would be considered an outgroup. As you notice, there's 33 differences with the black bear, 34 differences with the brown bear, and 40 differences with the polar bear. Like, this is different than the rest of those bears. And if you've ever done any studies on the panda bear, you're going to discover that it's actually more closely related to the raccoon. I made my students do this whole lab, and they were very upset with me when I told them that, and they discovered that, and they were just not happy. So phylogenetic tree actually looks more like a bracket almost. So here is the same question from the same year. It's just another question from that FRQ. And so they wanted them to say, okay, well, how long ago was the most recent ancestor of all the brown bears? And so if you follow the brown bear back, you say, oh, well, the most recent ancestor is right here. And so you drop that line down, you say, oh, well, it was, I don't know, 3.4 hundreds of thousands of years ago. And so you kind of use that to work with. Also, same thing, they may say, okay, which two could you rotate? Well, I can rotate the European brown bear and the polar bear because they share a node. I can rotate the Western North American brown bear and the Asian brown bear because they also share a node. So those could be things that are rotated. It also shows that the European brown bear is most closely related to the polar bear, and that the Western North American bear is most closely related to the Asian brown bear. Okay, so that gives you kind of a see of how they can use time, and then also a little bit about, rotating groups. And then we can also have traits put on it. Okay, so this is a trait chart. And so if you look here, you can kind of see that it's got different characters: lactose, protein A, protein B, casein. And you can see that the cat and the human have the first two, but they're missing the second two. And then you can see that the cow the pig, and the horse have all four of those different characters. And so they would give you a phylogenetic tree and you have to fill it in. Well, logically, since the human and the cat are similar, you would put them down here on this bottom. And then since the cow, horse, and pig are also similar, you can put them up here on this top one. But they want you to also include the characters. So all the organisms have lactose and protein A. So you would say that this must have taken place in that ancestor. And then casein and protein B must have emerged after the branch. After they diverged from one another, they must have, gained this mutation that allowed them to create the casein and the protein B. And so that's how we know that's there, because these ones have it and the human and the cat do not. So 710 speciation. I'm running out of time. So speciation is just the creation of new species. Okay, so thinking about what is a species, biological species concept states that two organisms are of the same species if they are able to interbreed and produce fertile, viable offspring. So they have to be able to have offspring together, and those offspring have to be viable. They need to be healthy, and they need to also be able to reproduce. So we have these things called prezygotic and postzygotic barriers. A prezygotic barrier, think about the words, pre is before, zygotic, of course, zygote, that's what's made when you have sperm and egg come together. So prezygotic is going to be something that takes place before your zygote is formed. Okay, so the first thing is a behavioral isolation. This has to do with the two organisms have a different mating ritual. There's different song, different dance, different something that causes them not to mate because they have different behaviors. The next one we have is temporal. This is where two organisms are going to mate at different times of either the day, different times of the year, different times of the month. Because they mate at different times, they don't mate with each other. Geographic. These organisms are separated by some type of geographical barrier. There's something that separates them from one another, and because of that thing that separates them from one another, they don't come in contact and they don't mate. Habitat ecological isolation is very similar to geographic. They just live in different habitats. Again, because they don't live in the same habitat, they don't come in contact with each other, thus they don't mate. Mechanical, the two organisms are incompatible anatomically. So this has to do with that their orientation isn't going to work, like they can't line up to mate. Yeah, I'm just gonna leave it there. And then gametic, this is where two gametes are going to be unable to fuse. So like that would be more like external fertilization usually, where we would have to make sure that, like, let's say sea urchin, that the sperm of one sea urchin isn't going to fertilize the egg of another species of sea urchin. So they have different glycoproteins, so the sperm does not fuse with the egg. So we don't, of course, make our zygote. And I never gave you examples. I'm so sorry. Behavioral would be like our blue-footed booby doing a dance. Temporal would be like the eastern or western skunk. One mates in the summer, one mates in the winter. Geographical could be, I don't know, the two different chipmunks that were separated by the Colorado River. Habitat could be a snake that lives in a tree or a snake that lives in the water. Mechanical could be two different snails, and I already gave you an example of gametic. Postzygotic, this has to do with after the zygote is created. So after we form the zygote, this is what's going to inhibit. So we've already interbreeded and we made a zygote. So now we're looking at something about it not being fertile or not being viable. Okay, so reduced hybrid viability, the hybrid is not viable, it's sick, it's weak, so it's not going to survive, and thus it won't pass on its traits. Reduced hybrid fertility, it's sterile, it's not fertile. So like a mule is an example here. A horse and a donkey are two different species, so when you mate them together, the mule, the hybrid of them, is not going to be able to also have more mules. And then hybrid breakdown. Hybrid breakdown has to do with the first generation. It's perfectly fine, it's viable, it's got no issues. It's going to survive, it's good, it mates again, and that's when we start to see the weakness, we see it feeble, we see that it starts to become sterile, like there's a breakdown of those hybrids. So this leads us into speciation, like how do we get different species? So sympatric speciation is where they're taking place in the same area. So our new species is going to be, from an ancestral species, it has the exact same geographical region. This is usually, we stop this by trying to have habitat isolation, behavioral isolation, sexual selection, and polyploidy. And so because of the fact that, this snake decided to go live in a tree, it no longer comes in contact with the other snakes, and thus it is going to not mate. And if you see the decrease in our gene flow, you see the decrease in these different individuals mating with each other, that's going to cause the two individuals to become different species, and they go into different populations. So there you see the hawthorn fly, and I cannot remember what the other fly is. But these are two different flies, and they picked two different plants. I thought one was a blueberry, one was a hawthorn, but I could be wrong. But since they went to different plants, they of course don't mate with each other. There's that habitat isolation. Allopatric has to do with that they're apart. The chipmunks that were separated by the Colorado River is showing allopatric speciation. So this is occurring where they are becoming isolated due to some geographical change. So here you see they're separated by the Colorado River. And then 7-Eleven, which is our last unit or our last topic, is on extinction. Okay, so in the Earth's history, there's been all these different mass extinctions. These extinctions usually come from some type of ecological change. And then a lot of our extinctions, not mass ones, but a lot of our extinctions are due to human activity. So the big ecological changes that can take place that can lead to extinctions, if anything's happening into their ecology, anything that's happening in their environment. So the mass extinctions were due to the ocean levels rising or falling. And so as the glaciers, released and they moved, that changed the sea basin, which changed a lot of the marine environments. Climate change, as the temperature increases or decreases, it's causing individuals to not be able to survive. Oxygen level of the oceans changes, and that's going to lead to decreases in different individuals. And then of course, as the atmosphere and the ocean chemistry changes, human activity has introduced different species are going to lead to changes. Maybe they become invasive species and they take over an area. That's going to lead to extinction of some of our native species. Over harvesting certain crops can lead to the extinction of certain crops, if there's a certain type of disease in it. So for example, the bananas that we all know and love, the Cavanache, whatever the banana they are, they currently have a fungal infection that's taking out a lot of the trees. And so we soon are not going to have bananas, or at least the bananas that we all know and love. And the banana flavoring that you all taste in things is actually an extinct banana. It was like the gross major, I believe. And that was wiped out by some type of fungus. It's already extinct. Burning fossil fuels is changing our atmosphere. It's giving us global climate change, pollution, overpopulation. All of that is leading to extinctions. So advantages of extinction is that it opens up opportunities for new species to emerge. Yay. It allows there to be, or new organisms filling in different niches, which is allowing them to change and allowing them to evolve. But that also wipes out the biodiversity. It also destroys existing ecosystems. It destroys existing kind of pathways and food chains and food webs and all these different interactions that the organisms have. It ruins those. And it's also a very long process to fill these new niches. Like evolution doesn't happen in a snap of a finger. It takes a little while. And so this also talks about how the amount of diversity in ecosystem is determined by the rate of speciation and the rate of extinction. Okay, so technically that is all the content. I have all these questions in there. It's not like it hurts me to keep going. So I'm going to keep on going. I'm so sorry that I've run over an hour. You're welcome to log off. But I did want to go over some practice problems in case you did want to stick with me a little longer. Don't forget, follow AP Bio Penguins, and I'm going live again next Saturday, 4 o'clock, same time, same place, over Big Idea 2, if I can get that PowerPoint put together. So let's look at a multiple choice practice question. So in this one, we're looking at a character table. So if you look at this character table, you can see the we're looking at the different traits. So if you look across, you can see that Y is not going to share any of the traits with the other organisms. And so you kind of like, okay, well, that allows us to eliminate B and D, because of the fact that you see here that Y is somehow closely related, and you see here that Y is somehow closely related. And so we know that that allows us to know that it's either A or C. And then you can see that, X and Z are very closely related, and then V and W are also closely related, which allows us to say, okay, well, that knocks out C, because now we see that, of course, our X and our Z are close, V and W are close, and then Y is our outgroup, which leads to A being your answer. So I hope that made sense. Here is the same one we saw before. I accidentally reused it. I didn't realize I did. But so they're asking us to create the phylogenetic tree. So we've been given this character table. So we say, okay, well, E. ferus and E. africanus have one amino acid difference between them. So we're going to put them onto our tree at the closest range. Notice that the kind of a smaller little V shape. Sometimes they really will focus on that, and sometimes they won't when they do these diagrams. But every single phylogenetic tree that I've ever seen has either been completely filled in for you, or they've given you just the template and you're going to add your words to it. So after we've had this E. farinus and E. africanus, you can say, okay, well, there's three differences between A. forestria and G. gallus. So you can put those onto the trait. And then, of course, our D. polypus is our outgroup, which has the least amount of differences. Now, notice there is a, what's it called? There's a note here. So you could have switched the A. gallus and the A. forestria and G. gallus here, and you could have switched these two. You also notice that there's a note here. You could flip the entire thing if you wanted to. So there was 4 different answers that this one could have been. The question also, I believe, asked us to provide reasoning for the placement of the tree of the one that's least related. So we put D. polypus there because it had the most differences between all the other organisms. And so you see, okay, there's the most amino acid differences cause those changes. And then B, so we're supposed to identify whether morphological data or amino acid sequence data is more likely to accurately represent the true relationship and provide a reason. Well, why would we want to pick amino acids over morphological? Well, amino acids are going to show the differences because of the fact that you can have a change in your DNA, and as you have those small change in the DNA, those are going to show they're more closely related versus having a similar structure. Similar structures could be due to convergent evolution. They could be analogous structures. They're not showing common ancestry, but they could be related. And so they actually let you have the point for either one on this. So you could have also put morphological data, and you could have said, well, because they had similar morphologies that could lead to similarities. Example could be like chimps and humans, how we have similar proteins, but we have different morphology. That word, sorry, y'all. So here we see these Malguppies, and this is my last practice question. Yeah, I think it's my last practice question. So here you see the Malguppies. We're looking at our error bars. The reason I put this here is because students oftentimes make errors when it comes to these error bars. So I want to make sure that we understand what's happening. So they're basically saying that we've now put them into a new place. There's no predators present in this group, and then there are predators present in this group. We want to see how the number of spots they have changes over time. So describe the change in genetic variation in population 0 versus population 6. And so you can look at that error bar. You can say, okay, well, it increased the number of spots. And you have been trained this year. Love your teachers, but you've been trained to know that if the error bars overlap, the data is not significant. That's not what they're asking you here. So the standard error of the mean is saying how much variance is there in your data. Okay, if you have a very big standard error bar, it means that your data is all over the place. It's not consistent. It doesn't, sit around the mean. Your data is just widely spread. And then if you have a small standard error bar, it means that your data is very concise, it's very accurate. It's like right, it's got that precision. It's like right there. Okay, so this question is asking us, how does my error bar change? Well, my error bar changed from being very diverse, right? There's a lot of variation to not so much variation. So that shows that we have a decrease in our genetic variation. And the reason why I said that is because the standard error of the mean gets smaller. Okay, so that tends to kind of trip students up a little bit. So I want to make sure that I did mention that. And then it says propose one mating behavior that could have resulted in the observed change between 6 to 20 months in the absence of a predator, which I clearly used the wrong diagram on that one. We're gonna use the one that goes downward, y'all. I'm so sorry. And so because of the fact that we have the absence of a predator, no, I'm sorry. Y'all, I'm reading this too fast. We were on the right diagram. I'm so sorry. We were on the right diagram. I'm just trying to rush to get y'all done. I'm so sorry, y'all. So as you see, the number of spots increased. Okay, why would the spots increase when there's no predator? The spots cause you to die. Duh. So because of the fact that we have no predator, there's directional selection that's going to allow for more spots to lead to, having more mates. So sexual selection for individuals with more spots, and then it could also just be random mating that increase the spots. Okay. Then we see we have the presence of the predator. the downward arrow, or the declining one. This shows that we have less spots when the predator is present. Well, less spots because, of course, they've been eaten. So there's your directional selection against the individuals with large number of spots, directional selection for the ones with spots, natural selection if you talk about how they had more spots, which caused them to be consumed, and thus we're seeing a decrease in those individuals. We're seeing increased individuals with less spots. They're more likely to survive, more likely to have more offspring, yada, yada, yada. So that is the end. So if you have any questions, you're welcome to throw them in the chat. I'm going to just do real quick information real fast. So if you're not following Macro Learning, be sure you're following Macro Learning. Be sure you're following me, AP Penguins. We're on day, like, I don't know, 7 of the AP reviews. So every single day I've been posting live on my Instagram page, doing practice questions to do each topic of the CED. Of course, follow Macro Learning on TikTok, and please don't follow me on TikTok. I'm cringy as cringy can be. And then, of course, hopefully you're subscribed to Macro Learning's YouTube channel. And then if you don't know, this is my website. So apbiopenguins.weebly.com. And so if you go here, there's a ton of resources for you. So this gives you access to a 351-page review document. There are game codes. So I've got codes that will let you get into almost like 80 plus review games. These ones right here are the exact same as the daily review I'm posting on my Instagram, and they all have explanations for them. I am not done. I still have one more unit to finish, but we're not there yet, so it's okay. There's also TikTok reviews. So I've got all the TikToks from like last year where I did random topics. And then I've been working through every single standard in the CED. I'm currently in the middle of unit 7, but if you want I'm currently working on them. The daily review has the PowerPoint for my daily review. Macro Learning Sessions has the PowerPoint for my sessions, plus any of the sessions that I've done with Macro Learning this year. And then there's a couple other things. If you want to get to any of my old ones, look, all of my previous reviews are there for you. So hopefully that this has been helpful. I do not see that there are any questions in the chat. So I hope that this was helpful. And remember, AP Bio Penguins are just a success. Bye, y'all. Where's my button? See, me and this thing just